Hello everyone and welcome to Chapter 21 screencast, which covers our cardiovascular system, specifically on blood vessels and circulation. We'll first start out talking about some of the functions of our circulatory system. We all probably already know that it's going to carry blood, and in that blood we are going to carry nutrients, waste products, and gases that we can exchange with our tissues. We can also transport hormones, components of the immune system, molecules that are required for coagulation, enzymes, and then of course we already have our nutrients, gases, and waste products. We are also going to regulate blood pressure, and we'll get into some of the details soon, and we can direct blood flow. Next, we're going to get into some structural features of blood vessels. Our arteries are not just arteries. We can break them down into three subcategories. We have elastic arteries, which are usually the larger arteries like the aorta. We have muscular arteries, which are a bit smaller. And then arterioles are little arteries. Then we are going to turn these arterioles into capillaries, which would be the smallest type of blood vessel that we have. And this is where we have exchange with tissues. Maybe we're giving off some oxygen, picking up some carbon dioxide and some other waste products. And then that blood will be put into our venules, which are very small veins, which will filter into our small veins, and then medium veins and large veins. Some differences that we're going to have between arteries and veins is that our veins will have thinner walls than arteries and contain less elastic tissue and fewer smooth muscle cells. So we'll look a little bit into the histology of what makes up these blood vessels to get a better idea. But before we get into arteries and veins, let's talk a bit more about capillaries. Our capillary wall is going to be the thinnest of all the blood vessels. It is made up of endothelial cells, like we see over here, which is really just simple squamous epithelium. Remember, simple means one layer, squamous means flat, and epithelium is the type of tissue. So it's so flat that its nucleus gets kind of shoved to the side, as we see over here. We're also going to have a basement membrane and a delicate layer of loose connective tissue to really give the capillary some support. We will also have some scattered pericapillary cells that are really just fibroblasts, macrophages, or undifferentiated smooth muscle cells. We will have substances moving through our capillaries, and by diffusion, they can leave or enter the capillary. So we can have lipid-soluble and small water-soluble molecules move through the plasma membrane. The larger water-soluble molecules are going to pass through fenestra, or gaps that we will find between the endothelial cells. And I'll show you an illustration to really give a good depiction of what fenestra and gaps we're talking about. Because there's not just one type of capillary. There are three main types of capillaries. First, we have the continuous type, where we have no gapping between the endothelial cells. There's also no fenestrae. By having a lack of these fenestrae and gaps, it makes it less permeable to larger molecules than other capillary types. So areas where we'll find these continuous capillaries are in the areas of muscle, skin, the lungs, and nervous tissue. When we look at fenestrated capillaries, you can already see that there are small holes here, which we call pores. And the endothelial cells have numerous fenestra. So the fenestra are going to be these openings right over here, and this is where we don't have cytoplasm here, but instead just have a thin plasma membrane with a porous diaphragm that allows certain substances through. So we say that fenestrated capillary are highly permeable. Areas we would find this are like in the intestinal villi, where we need to pull in nutrients. The ciliary process of the eye, the choroid plexus, which we would have in the ventricles of our brain, and glomeruli of the kidney, where the glomeruli are part of the nephron, the structural and functional unit of the kidney. And the last type that we have is a sinusoidal capillary. We are going to have a large diameter with very large fenestrae. And you can just see that this capillary kind of looks like Swiss cheese. We've got these large opening like spaces here. Areas we will have this type of capillary would be in our endocrine glands, like the adrenal medulla, where large molecules are able to cross the walls. Areas of the liver, bone marrow, and also the spleen. And this is a great illustration to show you the junctions of the endothelial cell. So here's one endothelial cell, and you could see that is tightly bound together here in a continuous capillary when it joins the second endothelial cell. Whereas here we have our fenestrated capillary. So I know in the illustration beforehand it just looked like one large hole, but we have areas where we have fenestra that have that thin diaphragm connecting the endothelial tissue a bit more together. And there are going to be areas where we have fenestra without a diaphragm. So it is an actual pore or hole. And lastly, here's our sinusoidal capillary, where we're going to have very large openings. This is the one that looked like Swiss cheese. And here we see a large fenestra between the two endothelial cells. There are some other more subcategories of capillaries, but these are a bit more limited throughout the body. So we have sinusoids that will have a large diameter and sinusoidal capillaries, which we will find in the liver and bone marrow. And then our venous sinuses are going to be similar in structure, but even larger. So we'll find these in the spleen, and we will give specific names to the venous sinuses in the brain in our AMP2 course. A bit more on our capillary network now. The way that blood is going to flow is first it's going through the arteriole here, and then it goes through a meta-arteriole, and then through what we call the capillary network. We can have blood flow through what we call the thoroughfare. So this is the main route of moving through to our venule. But what we really want is blood to flow all throughout this network so we can slow the blood down and really exchange our nutrients and waste products with the tissue. But there are times where we need to regulate the blood flow, so we can use instances in which our arteriole is going to vasoconstrict. The meta-arterioles can also vasoconstrict. Obviously, these can also vasodilate. And we also have precapillary sphincters that can contract to close off the lumen in order to keep the blood flow in one direction. So here is a little bit of a difference as to what is a true capillary. When we have true capillaries, that means that our precapillary sphincters are not going to contract, and we have full flow from the arteriole through the full capillary network, and we are exchanging nutrients and waste with the tissue cells. These can be considered like on and off ramps from and to the vascular shunt. A vascular shunt would be when blood flows through the arteriole and it directly connects to the venule, so that we have that one-way direction of the blood flow. This is also called an arteriovenous anastomosis. And just to give you a good idea as to what regions we're talking about, here is our true capillary region in which blood flow is going through all directions, and we are exchanging nutrients and waste products with the tissue. And then our vascular shunt would be this region right over here. And you could see located in these areas would be where the precapillary sphincters are. So this kind of encircles this vasculature right here. Here's another great illustration from your book in which we see our true capillary being used here. Blood is flowing in all directions throughout this capillary network. Over here, our precapillary sphincters are contracting, closing off blood flow to the network, and instead ensuring that it is going through the vascular shunt here, or the arteriovenous anastomoses. Now let's talk about the histology that make up the arteries and veins. The innermost layer is called the tunica interna, or it's also called the tunica intima. And like we mentioned when we discussed capillaries, we are going to have a layer of endothelial cells, or the endothelium, along with a basement membrane, and then we have our connective tissue layer, which would be the lamina propria. We'll also have a layer called the internal elastic membrane, where we will have fenstrated elastic fibers. The next major layer would be our tunica media. So here it is on the artery, and here you can see it on the vein. Immediately you could see that it is much thinner in the vein than it is in the artery. This layer will keep the smooth muscle cells arranged circularly around the blood vessel and will help the blood vessel vasoconstrict, where we are going to narrow the blood vessel wall, which will decrease blood flow. And we can also vasodilate, which would increase the diameter of the blood vessel, allowing the smooth muscles to relax while we vasodilate. And also we are going to increase the blood flow when we do this. And our very last layer is the tunica externa. And you could see that it is thinner in the artery than the vein. This layer is made up of connective tissue and can vary from either dense, regular connective tissue near the vessel to loose connective tissue that is going to merge with the connective tissue that surrounds it. So here's just a breakdown of the different layers. Once again, we have the tunica intima or tunica interna. Starts off with the endothelial cells along with a basement membrane and our connective tissue layer, which is the lamina propria. We have the internal elastic membrane, and after that we are going to get into the tunica media, made up of the elastic fibers and smooth muscle. Here's the external elastic membrane, and that brings us to our last layer, which is the tunica externa, or we can also call it the tunica adventitia. You can also see on top of the blood vessel, we have a nerve coursing through, because we have to give innervation to these blood vessels as well. Especially it's going to control the smooth muscle here and when we need to vasoconstrict or vasodilate. And these blood vessels here are called vasa vasorum. I pointed out these to you in a couple examples on the cadaver bodies in lab before, and we'll discuss these again. Here's a photomicrograph of an artery versus a vein. So you can see the much thicker arterial wall here compared to the wall of that of a vein. And we've nicely shown you the three different layers. The tunica intima, that would be closest to the lumen here, and tunica intima over here. You can see the thicker tunica media in the artery. Here's the tunica media in the vein. And lastly, the tunica externa or adventitia on the arterial wall. Next, let's go through the subcategories of arteries. We mentioned the elastic artery. These are also called conducting arteries. They're working to conduct their blood flow to the specific area of tissue that they need to go to. So these will be very large in diameter, and also they need to be able to withstand high pressures. And we see fluctuations between the systolic and diastolic pressure. We'll also have more elastic tissue than smooth muscle, which is why we call these elastic arteries. And that helps to kind of give more strength to these arteries that can endure that high pressure. Next, we have our muscular artery, and we can call these medium arteries. They will have the smooth muscle that will let the vessel regulate blood supply by constricting or dilating the blood vessel. Muscular arteries are going to have a pretty thick wall too. They'll have about 25 to 40 layers of smooth muscle. And we can also call these distributing arteries because the smooth muscle is going to allow the vessel to partially regulate blood supply to different regions of the body. We are getting more specific here. And if they are even more specific, we will call them small muscular arteries, and they will be adapted for vasodilation and vasoconstriction. The last type are arterioles. This is going to be the smallest type of artery that we have. And this is when we transition from the small arteries over to our capillaries so we can start exchanging substances. At this point, we can still differentiate the 3 layers or 3 tunics of our artery, and we will still see that vasoconstriction and vasodilation will take place here. Now let's discuss the subcategories of veins. So the smallest vein that we will have is called a venule, and their job is to drain the capillary network. The histology here is that tunica intima is made up of endothelial cells. We will still have the smooth muscle layer, but not quite as many layers as what we've seen before, just a few layers here. And our tunica externa would be made up of connective tissue. As the diameter of our venules increase, we are going to start to see an increase in the amount of smooth muscle that we find in the tunica media as well. The next subcategory are small veins. Here, again, we will see more smooth muscles within that tunica media as we increase the size of our veins. And our tunica adventitia or externa will be made up of more of a collagenous connective tissue. Our medium veins are going to go between our small veins and our large veins. Nothing to add there, except that once again, as we go larger in diameter, we will have a larger tunica media. And our large veins, we'll see that the tunica intima is thin, but we have our endothelial cells, a thin layer of connective tissue, and some elastic fibers as well. The tunica media will have those circularly arranged smooth muscle cells, and our adventitia is going to be our predominant layer, nice and thick here. Another thing that we need to mention when discussing veins is that they have valves. And we'll always find these valves in the veins as long as they are greater than 2 millimeters in diameter. The valves are made up of tunica intima, and they're just 2 flaps that are going to overlap so that as the blood moves through the vein, if it ever tries to go backward, the valves are there, or the flaps of the valve are there in order to catch the blood, and that closes up the valve. So that in our lower extremity, we can always fight gravity by being able to move the blood through and stop it from moving back down to our feet. And for that reason, we will always find more valves in the veins of our lower extremities than in our upper extremities. There are 2 different types of vessels that we need to mention. One of them you have already kind of learned when we discussed the full body veins, but you may not have recognized exactly what was going on there. And I'm talking about the hepatic portal vein. So here are the structures that I just mentioned. Here's our superior mesenteric vein that is draining up this way into the portal vein. This is our inferior mesenteric vein that is moving on up, and you can kind of see it back in here, draining into the splenic vein. And our splenic vein, which is draining the spleen, is headed over to our portal vein as well. So let's describe what's happening in the abdomen as we drain the venous blood. We are first taking the arterial blood that has the great oxygen within it and giving that supply to our stomach and our intestines. And we know that within the stomach and intestines, we have food and drinks that we have in there. And that capillary network here, the first capillary bed, is where we are going to give off the oxygen to the tissues, but we are picking up all those nutrients and possible toxins that we have taken in from the food and drinks that we intake. So then we move that back into the capillary bed. And so now we can call this oxygen poor or deoxygenated blood, but it's nutrient rich blood. And so we send that over to the hepatic portal vein, which then takes it into the liver, where we will meet our second capillary bed in the liver sinusoids. From here, we will give off all the nutrients and toxins that we have absorbed from the stomach and intestines to the liver cells or hepatocytes. We are going to try and detoxify any toxins that we have there and store nutrients that we need. And we are left with the low oxygenated blood that will travel from our hepatic vein into the inferior vena cava, which will take it back to the heart, of course. So just to tie that back into the picture that we have here, veins that we described, like the superior mesenteric vein, it's taking all of that nutrient-rich but oxygen-low blood, putting it all into the superior mesenteric vein at this point, carrying it up to the portal vein. Same thing here with the inferior mesenteric vein. We're taking substances from the colon, the rectum. Okay, all of this is filtering in to our splenic vein. All of the splenic vein substance is all going into the portal vein here, the hepatic portal vein. And remember, this is nutrient-rich blood at this point, oxygen low. So once we get into the liver, it's not shown in here, but it would go to the second capillary bed, and we would get rid of toxins, store nutrients, and then if we were to flip this liver down, we would have these hepatic veins that extend from it, which filter into the inferior vena cava, which will take it back to the heart. The second type of vessel we have is the vasa vasorum that I mentioned previously. These blood vessels are extremely small, and they are the ones that are giving blood supply to the vessels themselves. So you can see there's a couple different examples as far as how they are giving supply, but I just want you to know the general term vasa vasorum. Now, we've also mentioned that we need a nerve supply to our blood vessels as well. So these are going to be unmyelinated sympathetic nerve fibers that form a plexi in the tunica adventitia. That was our last layer of our blood vessels. And when stimulated, they will vasoconstrict. Our small arteries and arterioles are innervated to an even greater extent. And some of our blood vessels are going to be innervated by myelinated fibers that act as baroreceptors, which, if you remember from our chapter 20 lecture, they're going to monitor the stretch within the blood vessel wall and detect any changes in blood pressure. Unfortunately, as we get older, our arteries just aren't as durable as they once were before. We have two conditions here. We have arteriosclerosis, and this is kind of what I like to call an umbrella term because more specific pathologies occur underneath this umbrella. So we can use arteriosclerosis as just a general term for any degeneration changes that are going to take place in an artery, making them less elastic, usually due to age. The other term that we can use, atherosclerosis, that would fit underneath the category of arteriosclerosis, would mean that we have deposits of plaque within the walls of our artery. And of course, as more and more plaque develops in this area, we are narrowing the diameter of the blood vessel, and blood cannot flow as efficiently.